Okay, so I'm hoping everyone can see my slide in full screen mode. Thank you for the opportunity to speak to this group. I'm part of, as Jenny mentioned, we've been collaborating with the ClinGen SVI working group's computational subgroup for the past couple of years on improving, maybe sort of redefining, thinking about a little more deeply about specific criteria regarding the use of computational tools in clinical variant classification. And I'm going to start out by just sort of broadly giving a sense of what computational tools in this space do. Generally, I tend to think of these tools as being part of a family of tools that predict the impact of missense variants. This is broader than missense variants, but the talk is specific to missense variants. I'm just going to set it up, motivate all of this with missense variants, but it generalizes. And the idea is, given some information about a variant at the DNA level, extract some features that are related to DNA sequence, or maybe a little downstream related to protein function and protein structure. Combine these features into some kind of a model that would enable the classification of variants into two categories broadly, pathogenic and benign. And historically, the field has used different terms to describe these two classes. So there's deleterious, pathogenic could be synonymized with deleterious, damaging. Benign could be synonymized with neutral. They mean different things in different contexts, but for all intents and purposes, these tools have been used in these different contexts, and they've been interchangeably used. And I think, for this audience, I'm just going to say that there's two classes, pathogenic and benign. But just understand that impact is a very broad word in this sense. There's a rich history. There's probably over 100 tools that kind of accomplish this task using different combinations of features. Some of them rely solely on, let's say, conservation, for example. Some of them rely on structure function, as I mentioned before. Some of them integrate all these features using machine learning methods. Some of them are unsupervised in the sense that they don't use any kind of machine learning. They're just calibrated existing data sets. But in a broad sense, they all tend to output either a class label that says pathogenic or benign, or a continuous score distribution where scores at one end of the spectrum would be more indicative of benignity, and scores at the other end of the spectrum would be indicative of pathogenicity. In the context of clinical variant classification, these methods form one line of evidence, right? So they're basically integrated with other data sets. And I'm just going to broadly talk about these data sets. I think this audience is fairly familiar with the ACMG-AMP guidelines, but just to sort of recap, you can obtain, for a given variant, you can obtain information about its population frequency, some functional assay information, and basically also trace out its segregation history within families, and combine that and map these to different levels of evidence, different strengths of evidence, and the guidelines basically group these different data sources into these 4 lines of evidence, and they're twice over. So it's basically 8 months, one for pathogenic and one for benign. And I've just added a category in here called indeterminate, saying if a variant doesn't land in any of these 8, there's a 9th category. And the guidelines also provide some recommendations on how to combine these different lines of evidence to make clinical variant classification, to classify variants in the clinical context. So let's work through an example here. If I had 3 lines of moderate evidence for pathogenicity and one strong line of evidence for pathogenicity, I can now call that variant pathogenic. And in this manner, there are 5 different classes that a variant could land in: pathogenic, likely pathogenic, a variant of uncertain significance, likely benign, or benign. Computational methods in this context, as I mentioned, form one line of evidence, and they are generally grouped, they generally fall under these 2 categories here. So basically PP3 rule, PP3 criterion for supporting pathogenic, and BP4 criterion for supporting benign. These are, as you can see, supporting lines of evidence which are generally the weakest form of evidence. And the original guidelines basically provided some understanding of how, provided some indication on, how to use these tools, but they were not necessarily specific about which tools to use. And I mentioned before, there are hundreds of these methods. There's an inbuilt assumption that the tools are already calibrated for clinical use. And there's also one of the guidelines that also meant basically they present are that multiple lines of computational evidence should be used, and there should be agreement in order to be able to call a variant. So basically, in order to be able to use computational methods as supporting evidence for pathogenicity. But one of the things that I come from sort of the methodology development community. We develop methods, and one of the things that we know is that methods aren't necessarily calibrated for clinical use. So I'm going to illustrate this with two examples, one of polyphen-2 and one of our method, Metapredictors, because it's easier for me to describe. Usually when developers develop these methods, they do some kind of assessment or some kind of validation and fix thresholds based off of some objective measure of where the classifier performs well. So here is a table for polyphen-2 where there's false positive rates. In this case, the positive would be a variant being pathogenic or not. Essentially, polyphen-2 classifies anything that's at a threshold 0.902 or above as being pathogenic. And when you do that, as being probably damaging. And when you do that, the false positive rate that's expected is 10%. The same holds for a slightly weaker criterion, right? So it's possibly damaging. And the false positive rate is 20% of that in that instance. When we pick thresholds for our methods, we set it to essentially the 10% false positive rate and the 5% false positive rate. So very clearly, it's clear from this that first of all, the methods are not necessarily thinking about the 5-class classification system that ACM GMP guidelines have. And then they are also basically, the thresholds are basically decided within the context of the data sets that are used for the evaluative purposes. So the defaults may not necessarily work for clinical use. So we basically got together as a group and tried to address these questions. So when can a prediction be from these tools be considered clinically useful? What are the current practices in the use of these tools? And can we come up with a quantitative framework to revise these while being compatible with current guidelines? So these are 3 broad questions. And in the course of this, of course, we're, the group consists of people with different expertise and different stakeholders. So it was fascinating to sort of learn how all of these ideas could come together. One thing I do want to mention, and I'm not going to go too much into technical details today, but I do want to say that this is different from typical assessments that you would see, let's say, in a machine learning setting or a computational methods assessment setting, and it's technically challenging. And the reason for that is most of these methods, like I mentioned earlier, are trained to classify, basically they're binary classifiers. They're trained to classify variants into either pathogenic or benign. They're trained on data sets where the labels are usually disease or not disease, and that's about it. They output continuous scores, as I mentioned, and each tool has its own distribution shape across the space of all variants. Having to map this to 9 different categories of evidential strength is also basically another layer of complexity on top of this. So before I go into just a little bit more detail, I'm just going to stop and pause if there are any questions. If there are sort of big picture questions, I'm happy to take them at the end. Drop them in the chat. But if there's something that needs clarification, I think I'm just going to wait for a second. Okay. All right. So I'm going to jump into a little bit of detail just to illustrate what this effort entailed, right? So the first question we had to ask was, what does it mean to be supporting moderate and strong and very strong evidence? I mean, is there a quantitative definition for these? Fortunately, Taftikin et al. Had this paper about 4 years ago where they framed the ACM GMP guidelines in terms of a Bayesian classification framework and used odds path or likelihood ratios as ways to quantify these different levels of evidence strength. And odds path and likelihood ratios, in general, they're very routine and commonplace in diagnostic testing, to a certain extent in machine learning. So there's a nice one-to-one correspondence between how one would typically evaluate methods and this particular term here. So again, without going to the details, we have a way where we can basically map these 4 strengths for pathogenicity and 4 strengths for benignity to some numerical value, the likelihood ratios here, 350, 18.7, 4.3, and 2.08. But the next question that you could ask is, when we talk about a Bayesian classification framework, obviously, whenever you think Bayes, you think of a prior probability, right? So in this work, the prior probability was basically estimated. It was sort of guesstimated, and it was a theoretical number that was chosen, and it was a reasonable number to choose. But we asked those questions: can we estimate this from real data? And for this, there's basically there are methods out there that kind of use ideas from positive unlabeled machine learning to be able to estimate these priors from a reference set. And in this work, we used Nomad, because it's variance coming from the population, as a reference set to estimate priors. So what happens as a consequence of this is that when we estimate the priors, our number is 4.4%, which is a little lower, which is lower than the 10%. It immediately bumps up the strength level. So instead of having the number be 350 for very strong, it's 1,124, and everything scales from that. What I also want to sort of, for the rest of the talk, what will be informative here is that there's a way to go from likelihood ratios to posterior probability of pathogenicity and benignity. And I'm going to use these terms interchangeably. So the point of this slide is basically just to say that you will see pictures and results in the context of posterior probabilities, but there's a one-to-one mapping with likelihood ratio as well. And finally, now that we have these different ideas come together into a theoretical framework, how do we translate this into actually estimating score thresholds and intervals and sort of giving guidance on how computational predictors can be used? And in our work, we basically had to define a localized version of this likelihood ratio, and we proposed an approach to calculate this. Just to give you an overview, take the example of a single tool here, and on this axis, I've just basically, let's assume we've organized all the variants and on an increasing scale. So for this tool, let's assume a value of 0 is benign and a value of 1 is pathogenic. Arrange these scores in ascending order, and just ask where do the variants land. And this is, again, assuming we have some kind of gold standard data set. So the red label and the blue label are coming from, let's say, in our case, we use ClinVar. We then take each unique score and then calculate the fraction of pathogenic variants around a small window, so a localized window here. And then we keep doing this for overlapping windows. So for each score, we get some notion of a posterior probability. And we do this starting from the top, going all the way down. And ultimately, for each score, we get a posterior probability value, and we just plot this. Now, in the table I showed you before, there are posterior probability thresholds that we can use to define very strong, moderate supporting. So there's these 4 lines here, and wherever these curves intersect with these lines, we can start now saying, okay, that's the particular score we would choose as our threshold that would ensure that we would meet those evidence strength criteria. You can do the same for benign, but you just go the other way. You'd start from the lower score, working your way up. And ultimately, this is what we were hoping to achieve, right? So we basically want to divide all these variants in this box to these 9 different intervals in a rigorous and quantitative manner, an objective manner. So for this task, we extracted data from ClinVar. We went through some filtering steps. We made sure that we didn't include any variants that were of uncertain significance, had at least 1 star for their submission status. We removed any variants in genes that contain only benign variants. So basically, we wanted to only include genes that had both pathogenic and benign variants. And then we also filtered against the training sets of the tools that we assessed. So for this assessment, we picked 13 tools and evaluated 13 tools. Some of them don't use machine learning and don't use training sets, so we didn't have to do any kind of filtration. But of those that did use training sets, we made efforts to remove variants in our data set that overlap with the training sets to address any kind of any possible biases. And we focused largely on rare variants, and our data set was split into these 6 groups here. For the purpose of this analysis and the rest of this talk, all of these would be sort of lumped in together as pathogenic or likely pathogenic, and all of these variants would be lumped in together as being likely benign. So they're just 2 groups here. So let me give you an example of one posterior probability curve. So this is for Revel, and I'm going to walk you through what's being plotted here. As I mentioned before, on the x-axis is the score, on the y-axis is the posterior probability. There are 4, the horizontal lines represent 4 posterior probability cutoffs corresponding to the strengths. So very strong, moderate, supporting. The black curve is the point estimate of the posterior probability of each of these scores. And the gray curve is the one-sided confidence interval. And the side or the direction for the confidence interval is chosen based on being more stringent. So for benign, it's going to be on the left, and for pathogenic, it's going to be on the right. And when we pick thresholds, we use the gray curve to define thresholds. I should also mention that Revel assigns scores between 0 and 1, with 0 being benign and 1 being pathogenic. And if you notice here, Revel does pretty well, right? So there's a curve that goes across the supporting line at around a little over 0.6, the moderate line close to 0.8, the strong line at about 0.9, and that does not meet very strong for pathogenicity. For benign, the curve touches very strong at the top end over here, and then keeps dropping down. At 0.2, we hit moderate. I missed strong here. So at very close to 0, we hit strong. At 0.2, we hit moderate. And then at around 0.2, 0.3-ish, we hit supporting. And, in the cases where we have curves crossing the line twice, we always pick the more stringent threshold. And for this, this process is fairly automated. We have an algorithm that kind of picks this out for a bunch of tools, and we try to avoid having to eyeball this. Now, let me show you another example where this does not necessarily happen, right? What happened with Revel. So CAD is one example, and this is not unique to CAD. I just picked CAD because it kind of makes the point. So here, what happens with the score is that after we hit moderate, the posterior probability flattens out and never hits strong. And the score thresholds basically are also different from what's used as the default, and I'll come to that in a little bit. So typically CAD 20 and CAD 30, generally CAD 20 is what's used to define a variant as being pathogenic or supporting evidence for pathogenicity. And clearly here, from this picture, you can see that 20 sort of gives you evidence for benignity, but not necessarily for pathogenicity. And for pathogenicity, you need something a little more stringent, and 30 would sort of get you there and would be able to give you moderate evidence. Are there any questions at this point? Okay. So I would like to make one note about the developer recommended thresholds. I'd mentioned this before. We took 3 examples. We could repeat these for other methods. And we just picked 3 examples that probably were representative and typical to the field. SIFT has a threshold of 0.05, and note that SIFT has an inverted threshold. So if you're below 0.05, you're pathogenic or disease associated. Polyphen 2, I already mentioned, is 0.902. I picked the more stringent threshold, and CAD is 20. And again, this is, note that this is not, in some of these cases, these are hard, they're built into the tool, and in some of these cases they're not hard recommendations, but they're ballpark recommendations made by the developers. And none of these actually meet the threshold for even supporting evidence. So this is just sort of to drive home the point that these methods are not necessarily calibrated for clinical use. There are other thresholds, as we saw in the previous picture, that will get us to supporting and moderate, but the default thresholds aren't necessarily the ones. So as I mentioned, we did this for 13 tools, and I'm just going to highlight a few key points here. In general, methods can provide more than supporting levels of evidence. Computational methods can provide more than supporting levels of evidence for both pathogenicity and benignity. Some tools can provide up to strong, and there are 4 tools that provided score threshold, 4 tools that had scores above which you could call a variant as being strong. Having strong, basically you could use the method as strong evidence for the variant being pathogenic. Just to walk you through this, for the moderate, I'm basically presenting intervals here, right? And so in this case, it's basically if you're at least 0.5, base del can give you strong evidence for a variant. But if you're less than 0.5 and at least, basically if you're at least 0.27 and less than 0.5, moderate, so on and so forth. And on the other end of the spectrum, there are tools that can give strong evidence for benignity as well. And they're not necessarily the same tools. So one case in point here is CAD. But CAD gives moderate evidence for pathogenicity, but on the benign side, you can actually get strong evidence for benignity. There is a score threshold that will give you strong evidence for benignity. We also sought to validate our score intervals, right? So now that we have these, we decided to wait. Well, the course of the project took time, and then we basically waited a year to let new data accumulate into ClinVar and asked, now that we have these thresholds and we apply these thresholds back, do we get the same expected posterior probabilities and likelihood ratios? And the procedure is pretty much the same. We just made sure that this data set did not have any variants from ClinVar 2019. But we did the same steps where we filtered with the training sets of the different tools. We made sure that the allele frequencies, and we're not including any common variants. And the data set numbers are down here below. It's a smaller data set, but serves the purpose, at least being able to validate and just test our method, make sure that everything makes sense. So here what I'm plotting are basically the 9 intervals, with the gray box being the indeterminate interval. The 13 tools are the rows, and each cell represents the likelihood ratio within that interval, calculated within that interval. So if everything was done right, we should see likelihood ratios that are at least the same as the ones I showed in the table before. So for supporting, it would be somewhere a little above 2. For moderate, it would be at above 5.3, and then for strong would be somewhere above 33. And overall, that seems to be the case, right? So the likelihood ratios in the ClinVar 2020 set are within expectation. And I just want to make a point that, tools behave differently than, even when you change data sets, right? It's not that these numbers are super close to the numbers that I presented earlier. They're much higher in some cases. As I mentioned, CAD does not provide strong evidence, mostly provides just supporting and moderate. But the other point I want to make is we also applied these thresholds to a data set from basically our nomad data set, the reference set. And we just want to ask, what are the fractions of variance that fall into these different intervals in nomad? And I've picked these tools, Basedel and CAD, just to sort of illustrate that generally the choice of tool involves a trade-off between evidential strength and false positive classifications. So for example, with CAD, you can maybe classify 24% of variance on average, 25% of variance on average as having at least supporting evidence for pathogenicity, but the best you can really do is moderate. And we also know that in the nomad set, we shouldn't be expecting to see 10%, because these are much higher than even the priors that we estimated, right? So what you'd like to see, so the priors that we estimated about 4%, 4.5%, what one would like to see really is the strongest the strongest level of evidence for a given tool would sort of land within that prior probability range. That's what would seem reasonable in this case. All right, so before I go into some of our recommendations and our suggested updates, I'm going to pause again for questions. I know that people have the ability to drop questions in the chat at the end, but if there's anything that needs to be addressed, I can address it here.
Hi, it's Jenny, Vikas. So there is a question in the chat, and I think this is a great question. Do you have a way to see if the ClinVar classifications were reached without considering PP3 and or BP4? So was it different when only looking at 3-star classifications? So I guess these are two separate questions. So the first, like, could you do this analysis independently of the in silico data?
So I can, I'll address this in a little bit. I mean, it is one of the caveats to what we're doing. We haven't necessarily looked at data sets. That information is not really straightforward to extract from ClinVar. I know that ClinGen has a repository of evidence, curated variants with evidence and what evidence was used. It's not very straightforward to extract, what variants used PP3 and did not use PP3. The assumption here is that historically, because it's been used as computation methods have been used as supporting methods, we expect it not to have a huge impact. To the last question, we've not looked into only 3-star classifications. I think here, from the outset, we were hoping to get as big a data set as possible, and I don't have a feel for the numbers yet off the top of my head, basically, but I believe that looking at only 3-star classification would drastically reduce our data set, and it might be challenging to do this on a small data set. But yeah, these are perfectly valid questions to ask. Yeah, we've been thinking about this too.
And I see that Sean made a comment to you earlier. This point that developer recommended thresholds do not meet even supporting evidence cannot be overemphasized. It's a really important result. So just want to make sure everyone had seen that too.
Yeah, I should also say, and I'll bring this up again in the end. Many of the members of our group are on this call, so and there's different perspectives, and we'd be happy to have a discussion and take questions also at the end. Have it be more open-ended. All right, just to sort of back up, this table here basically compares two sets of recommendations. So the original recommendations in the ACMG paper and what we're proposing. I'm just going to run through the key points. The paper is up on bioRxiv. I should have mentioned that earlier. For methodological details, for details in these recommendations, please feel free to take a look at the paper. In general, as I mentioned, computational methods were considered to be supporting evidence. We think that they can provide more than supporting evidence, provided they're used at the appropriate thresholds. We have to make shifts from the default. There were no specific recommendations for which tools to use. We recommend that tools that reach at least strong evidence for pathogenicity and moderate for benignity be used. But of course, our recommendations based off of this trade-off between false positive classifications and evidential strength. It is valid to use any tool as long as it is used at the evidence strength indicated, and it should be chosen before seeing scores and preferably any other evidence, right? So you want to make sure that you're not biased by the outputs of those tools before you choose the tool. I already mentioned that specific score thresholds can be used because we've developed a way to calibrate these to for the clinical use case. We did not systematically assess what impact combining tools would have, essentially using, let's say, a SIFT and a CAT and a polyphen. I mean, we did try just for that one combination, basically that triplet of tools. We just asked if using the consensus was going to get us more than supporting, get us supporting more than supporting, and the general results were not very positive. So essentially, we recommend the use of a single tool rather than an uncalibrated consensus. And I should mention at this point that calibrating multiple tools simultaneously in the way I described over here in this talk is a whole project unto itself, because now you have to start looking at pairs and triplets of scores and trying to find, now instead of having a posterior probability curve, you have more like a multi-dimensional plane, so to speak, hyperplane. From a decision-making standpoint, we say that evidence can be counted at the appropriate extent depending on the tool and the score threshold chosen. But again, the maximal strength must be committed to. So you can't take CAD and say, if it's moderate, I'm going to count it as strong, right? I mean, the max it can do is moderate. And the first point again has to be repeated here is that the selection of tool has to be done much before variant is considered or any other evidence. Ideally, any other evidence is viewed, but at least the choice of tool must be picked before seeing the outputs of the tool. And our work, we touch upon this a little bit, but it is clear that these methods are not necessarily independent of the other evidence code. So I should have mentioned, I did kind of allude to this early on, that some of these methods don't exclusively use conservation. They use, let's say, functional information. There's some overlap with other evidence codes, and for that reason, we make a recommendation on at least one overlap that we think is obvious. It's between PM1 and PP3. And PM1 is the mutational hotspot, so the presence of the variant in a mutational hotspot. And our suggestion is to, whenever these two codes are used in combination, the combined evidence cannot be more than strong. It has to be limited at strong. So just to sort of summarize and go over some of the key takeaway messages, as Sean mentioned, default developer recommended thresholds are not necessarily calibrated for clinical use. Computational methods can provide more than supporting evidence and even strong evidence in some cases for both pathogenicity and benignity. Single tool must be used in selector before seeing prediction results, and preferably other lines of evidence to avoid potential biases. And one thing I'd like to say is that our updates now, and to the question that came up earlier, motivate the need for more rigorous documentation of evidential lines in the future. You know, up until now, the assumption is that these methods were supporting, but now by introducing these new levels of strength, it's likely that they'll be more actively used to classify variants, and it would be important to document specifically what tools were used, what thresholds were used, and where the variant landed in terms of the strengths. One other aspect is what we propose here is a new approach to assess and calibrate predictions for clinical use. Like local posterior probability, in theory, can also be used for other continuous outputs. So maybe from high throughput functional assays, there'd be some tweaks that need to be done, but it's not that far-fetched to think that this method would work even outside of the use of computational methods. With sufficient data, this could be extended to gene-specific or disease-specific context too. But like I mentioned, dataset size would be important. And some of the caveats, gold standard ClinVar annotations often incorporate prediction methods. And you know, as I mentioned, there's no straightforward way to account for this, in at least at scale. Maybe for specific genes and variants, might be able to do it on a smaller data set. And there's definitely overlap between information used by predictors. And we've not, you know, we've addressed that in some cases, but not necessarily systematically studied this and addressed this. And one point I'd like to make is about the technical aspects, is that there are challenges in the estimation, right? So you know, a lot of this depends on, you know, what data sets we've chosen, you know, how big they are, how representative they are of how these methods are eventually used, estimation of the prior probability, some of the limitations of the rules and Bayesian framework itself, right? Like the assumption of independence between the two lines of evidence. And we know, at least from the computational perspective, that, you know, the output from a computational method may also be linked to other evidence codes in some ways. And we've developed this only for use within the ACMG AMP guidelines, and you know, this whole framework may not behave as expected in other contexts. So with that, I'd like to end and just acknowledge all the members of the computational subgroup, ClinGen PIs, and other collaborators. This was truly a team effort, interdisciplinary. There were bioinformaticians, machine learning, data scientists, clinicians. So it's actually one of the most exciting projects I've been a part of in a while. And I'd like to thank everyone for, first of all, thank you for the opportunity to speak, and I'd like to thank you for listening. And I put in the preprint information here and open for any questions and discussions. I see the chat is pretty alive right now. So.
Yes, thank you so much. And yeah, there's been a lot of discussion in the chat, so maybe we can go through some of these points, but this was really terrific and a great analysis. And I'm really excited to see so many members of the SVI on this call as well. So welcome everybody. So maybe we just go through some of the points in the chat. So one was how many genes were tested when you were setting those thresholds, and whether you observed any statistically significant difference between different genes for the different tools.
We haven't done a gene-specific analysis, but I think it is something that we hope to do. It again hinges on availability of data. And in this context, to the first part of the question is how many genes? So this whole data set, I should have mentioned up here. I don't know if it's, maybe it's better for me to just, so the numbers in the parentheses here are the number of genes that were tested. So I did not mention that when I was presenting. So just as an FYI, so for ClinVar, it was about 2,000 after we did all our filters. Let's go back to full screen. Yeah. Does that answer the question? I mean, I guess, you know, if okay.
Yes, I believe so. So, and obviously this was just on the total genes, so no comparison between different genes or different families of genes.
Yeah, I mean, families of genes is an interesting proposition because that's one way to make sure that we have enough data to be able to do this. So some of the technical details I didn't get to regarding the method is that we need in order to be able to get a reliable approximation of the posterior probability, we need substantial number of variants within each window here to be able to do this. So we set the cap at 100. So we need at least 100 variants to be able to calculate this fraction and be able to get the posterior probability and plot these. So, you know, for genes where we have that level of data, where, you know, we're talking about maybe, I don't want to give a number, but let's say hundreds or nearly, you know, to the thousands of variants, that would be possible to do. But gene family is an interesting sort of compromise, right, where you can combine data from multiple genes and be able to estimate these. But in principle, they should, you know, everything should stay the same methodologically, except that we'll have to re-estimate the prior for that gene or gene family, and then all of this holds, the protocol holds from there.
That's great. And then there was also some discussion about using functional data to kind of support the accuracy of the prediction. So do you have comments on that? Would functional data be necessary? I see there was some discussion in the chat, but
yeah, I'm happy to take it, but I know that Sean and Les have been sort of jumping, you know, I think, you know, they're, in general, to the question, the person who asked the question, their point, yes, we don't think of functional data as the gold standard truth. Calibrating against functional data may not be ideal in this case. But I think I alluded to this. So Sean's raised his hand. Okay, all right, Sean, I want to just let you take the floor.
So there's a really important point to make here, which is that in the end, the classification scheme is, you know, the whole classification scheme is meant to classify the sequence variants for their effect on human disease. And the data that are most relevant to that are measures of human diseases, things like penetrance and odds ratio. Those data, penetrance and odds ratio being almost direct measures, don't need to be calibrated very much. On the other hand, things like computational tools and functional assays, the output of a computational tool and the output of a functional assay have to be calibrated relative to human pathogenicity. This being the case, it is a very, very bad idea to try to calibrate computational tools relative to functional assays or functional assays relative to computational tools, because those calibrations become unlinked to human pathogenicity. And so the rule of thumb is don't do it.
Yeah, I just to add to that, one could think of a way to think of extending this approach to, instead of having this y-axis be scores from a predictor, it could be scores from a functional assay. But there'd be slight changes to the way we do the calibration, because now functional assays are bidirectional by definition, right? So you have variants that impact function, that have no impact on function, that have, that lead to reduced function or lead to increased function, right? And both those directionalities, I mean, that's a little different from benign and pathogenic, and basically this one, basically this increasing scale or decreasing scale. So the calibration procedure would change a little bit, and it's something that's certainly thinking about, but it's like, the basic tools or the basic steps would be extendable to functional assays, because it's just a continuous output. You could think of it as, hey, here's a continuous output from a method. But the output itself, the nature of the output is very different from what most computational predictors provide. So it would require some more thought to adapt to functional assays.
And there was also some discussion about using PM1 and potential for double dipping. Do you want to comment on that a little bit, and the recommendations for dealing with that?
Yeah, I think I'm going to, for that, maybe I'll just open the floor to Sean and Les, because I know that they've been thinking about this a little more deeper than I have. And as I mentioned, this is a team effort. So if either of you want to jump on, it's fine by me.
Great, thanks.
Go ahead, Sean. You answered it in the chat. You want to expand on that?
Unfortunately, I was chuckling at Les's response to a question from Julie Boyle at just the moment that this question was asked. So I don't.
Oh, he's just talking about adding PM1 on top of PP3.
Okay, so many of the individual computational tools that are built into the Metapredictors that Vikas was calibrating here actually use data that should be regarded as PM1, along with data that should be regarded as PP3, if there's even any distinction between the two. And consequently, this calibration combines some evidence that one might call PM1 with evidence that one would call PP3. And consequently, in the manuscript, we put a ceiling on the strength of evidence from the combination of PM1 and PP3 that one should be allowed to apply. And that ceiling is strong, or in a point scale, 4 points.
Thanks, Sean. And I should also add here that in the case of PM1, it's subtle, and that's why we had to do this. There are other evidence codes that overlap. For example, with Basedale, Basedale has two modes. Basedale can be run with allele frequency information provided and can be run without allele frequency information provided. And here we use the one without allele frequency because there's already a rule and combining and basically combining, there's a criterion and combining rules with respect to allele frequencies in the population. So in some cases, it's actually very straightforward to hold that out and make a recommendation. But in the case of this overlap with PM1, it's hard to tease that apart, and we had to kind of propose a workaround.
I think another general point that's worth emphasizing here is that, if you look at that table with the predictors that performed well, there are four, depending on any count, 4 or 5. And what's interesting about them is the good ones are essentially all meta-predictors. And they're using some of the other non-meta-predictor input as well as some other considerations. And you can actually think of these meta-predictors as conceptually doing what Richards et al. Recommended in the first place, right? Which is looking at multiple predictors, combining them in a thoughtful way and coming to a conclusion, except of course, these Metapredictors are doing it in a quantitative, objective way that's much more logical, rigorous, and systematic. And so it's not too surprising that what we're ending up here doing is validating the Metapredictor concept, which isn't that far off what Richards et al. Actually recommended, and making it much more uniform and consistent. And I think the problem with the Richards et al. Vagueness, and no criticism of Richards et al. It's a stupendously awesome paper that put this entire field on the right track. But that one recommendation had some work that needed to be done on it. And this is showing where you go with that. And by switching to a Metapredictor, you basically can do in a rigorous and systematic way what Richards et al. Recommended, and be prevented from that incredible temptation of, ooh, I like polyphen and SIFT and Fathom from this variant, and ooh, look at that variant. Looks good with GERP and MPC and Philo-P. That's the last thing that we should be doing. And so one of the big recommendations we're going to make, as Vikas said, pick a predictor before you know what the answer is for your gene, your disease, and stick with it and be consistent. And that leads to the most robust and consistent way of doing things.
Thank you for that. There was a question, which I think is a great one and probably on many of our minds, but how are these recommendations going to impact the current variant curation expert panels, and how do we incorporate this into our specifications moving forward? So I just wondered if you had any thoughts on that.
So one thing that I should have probably mentioned in the presentation, it's in the paper, is that for specific genes, if there are tools other than the ones that we've looked at here that have been calibrated in some fashion to basically classify variants in those genes, I think defaulting to those is reasonable. But we're in the context of genes for which such efforts haven't, and such as those from the VSEPs, right? But in cases for genes where there are no VSEPs, I think this would be probably the most, defaulting to these thresholds, and one of these tools, or the more generic approach that we have here, would be reasonable to do. But in terms of actual impacts on individual VSEPs, again, I think I probably would defer to Les on this one.
Yeah, it's what Vikas said earlier. It's always going to be a trade-off between large data sets that give you a precise answer versus narrowing it to a gene where you have a very small data set. And I think, and Sean, correct me if I'm wrong on this, I think one of the recommendations we discussed informally is that if you pick one, if you like one of these tools in general, SVI tends to like Ravel, and Ravel performs as described here. You look at your individual gene, and if for your individual gene, you're not seeing significantly anomalous behavior of that tool with your gene, then these thresholds are probably good for your gene because they're based on much more data than just your gene. But if you see significantly anomalous behavior, you might want to think twice about using a tool like this on your specific gene. Did I summarize that correctly, Sean?
Les, that is correct. And I think that the question to be resolved in the future is, if you see anomalous data, anomalous results, how much data, how much strength of evidence of an anomaly would you really need before it's reasonable to switch to either change the calibration for a specific gene, or move to some specialty tool for the specific gene, or I think sometimes it'll come down to specific domains rather than specific genes. And I don't know the answer to that question. I'm just posing it.
I can give you an example. We did, and Vikas obliquely referred to this earlier, we did a calibration of Ravel with a truth set of variants for RYR1 in our VSEP. And then this analysis was done later, and it's not that far off. It just really isn't that far off from the estimates that we got. So that means that that gene is performing pretty well, and probably that VSEP ought to consider switching to these thresholds because there is no evidence for anomalous behavior for that gene.
I think I saw Stephen raise his hand. Stephen Brenner.
I was just going to add to what Sean and Les said, noting that, so we would expect that indeed, most of the time for most genes that the weight of data coming from this large scale analysis would be most valuable, but that VSEPs do have also extensive expertise, and that special expertise may also allow them to better interpret limited data than what can be done in an automatic way as we're doing here.
Great, thank you for that. So yeah, it sounds like, you know, different VSEPs, I mean, once you have some data, maybe as a result of the pilot study or something, sort of looking at this and seeing how these thresholds hold up for your particular gene would be good. There's another question too about inheritance patterns. So for missense changes causing autosomal dominant versus autosomal recessive disease, should you consider different thresholds?
Yeah, we haven't looked into that. We haven't separated the genes. I think there was also a related question someone asked up regarding when we were using nomad variants as controls, do we exclude variants from AR condition genes? So what we did basically here, again, we didn't separate genes out by inheritance pattern, but when we did pull information from nomad, we still use the same set of genes that we started with. So when I say we use nomad as a reference set, I should be a little more specific and say that we didn't use all of nomad. We used variants from genes, only variants from genes that were in ClinVar as a starting point, right? So basically clinically relevant genes are the only ones that were included. But no, we did not look into separating by mode of inheritance. And I don't have a feel for whether it would be different thresholds or not. I think that's a good question. And I mean, I know, for example, one of the tools here, like MPC, for example, was developed exclusively for autosomal recessive disorders, I believe, or autosomal dominant. I can't remember. Maybe Anne, if she's on the call, could jump in and clarify. But you know, the MPC's performance is very different, if you notice, from the other tools, because it seems to lean more heavily pathogenic, and it's not necessarily great on the benign side. So most of these tools were developed regardless, in a more generic sense, without attention to mode of inheritance. So we, at least for the first version of this analysis, we haven't considered mode of inheritance. I don't know if that answers the question. But it's a good point. Maybe it's a potential future direction to consider.
Thanks. So are there any other questions? I know, I see another one just popped up. Oh, it's a thank you, Vikas. Any other questions? I know there's been a lot of discussion in the chat. I hope I didn't miss anything, but please feel free to, we have a few minutes left. So if anyone has additional questions, please feel free to speak up.
Dimitri has a great question.
Dimitri, would you like to? Oh, the points-based system at BP4. Dimitri, do you want to ask your question?
Yeah, So essentially the question is, there are of course a bunch of related questions. I'm sure people have a bunch of other questions to think about it, but one of them is point-based system when likely denial start from either -1 or -2. That essentially means you can have situations you have no evidence with the exception of computational evidence. And then it would look like you can get in a situation when supporting computational evidence just from point perspective, which seems to be enough to call something likely benign. And I think that's something that we really need to carefully think about it. And another related issue, which was briefly discussed before in some other communication, we need to have some recommendation what to do when evidence are conflicting and pointing in different directions.
Yeah, and I think overall, one of the issues that we've had a lot of discussions about with Richards et al. Is, as Vikas outlined early in the talk, it was necessary for practical purposes for the Bayesian point-based system to set the prior as 0.1, respecting what Richards et al. Said. However, you all know that if one is interpreting an exome, it is not the case that 10% of the variants in an exome are pathogenic, because that's what that is saying, right? That's the prior probability of pathogenicity in that person's exome. And so in fact, the prior probability is currently arguably too high. That leads me to answer your question, Dimitri, to say, I feel very comfortable with a piece of in silico evidence alone, even at the supporting level, pushing a variant from low VUS to likely benign. Not everyone agrees with me on that, and there's more work that needs to be done overall at balancing the whole system so that it optimally matches the observed prior and the observed posterior probabilities in the real world. That's going to take some work.
So just in the last minute or so, any other last questions? And I did want to thank you all for your input. This is just such a great topic. I'm sure it's one that we could talk for a lot longer. So thank you so much.
Thank you so much. I'll echo everyone's sentiments from the group that these were great questions, a great audience. Thank you.
I think everyone should give a big hand to Vikas, who made a great presentation today.
Yes, thank you so much. Great. Well, we're just at the top of the hour. So thanks again, everybody, for joining and for all the great questions and discussions, and to this SVI group for the guidance. We're always very grateful for all of the input and help. So thank you all. And I will share the recording with the listserv.